Hello, my name is Mollie Uhrig, and today I will be telling you a little bit about chromatin immunoprecipitation sequencing, also known as ChIP-seq. So ChIP-seq is a very nice method to determine where proteins are binding to DNA across the genome. So recently this has been used to also show where nucleosomes are binding on the DNA and where certain histone modifications are along the DNA, and they map this to the genome. So they can show where nucleosomes and histone modifications are happening across a reference genome. There are many pros and cons to ChIP-seq, so it actually is one of the older methods that is used to show protein association with DNA. There's some pros and cons to this, which I will discuss later. And actually, the development of many new techniques such as ATAC-seq or MNA-seq are actually preferred because they have greater capability with less background and less artifacts. So next I will tell you a little bit about the steps, the procedure for ChIP-seq. So first you need to cross-link the proteins to the DNA so that in your sample you have proteins stuck to the DNA right where they're binding. You do this with formaldehyde, which tends to be a very harsh reagent, so you can have some artifacts from treating the cells with formaldehyde. Next you need to lyse the cells and then fragment the DNA by sonication. So typically you want to have between 200 and 600 base pair lengths of DNA. This tends to be a pretty standard length, and this is one step of the procedure where you need to be a little bit careful so you don't over-sonicate and have shorter fragments of DNA. Next, you need to use an antibody to detect your protein of interest. So this will specifically bind to proteins that you're interested in, the proteins that are cross-linked already to the DNA. So this is considered the immunoprecipitation step. So after this, you use magnetic beads to capture your antibody protein DNA complex of interest. And then to isolate this, you wash and remove other proteins in DNA, so anything which is not stuck to your magnetic beads. This helps to get rid of excess signals that are not of interest so that you are only getting the signal that you're looking for. Then you need to reverse these cross links and get rid of the protein and isolate the DNA only. So to do this, you can use RNase digestion and protein digestion with proteinase K. This will leave you with only the DNA, so you can then sequence this DNA using next generation sequencing and map it to the reference genome so that you can see exactly where in the genome these proteins have bound to the DNA. So how is this relevant to modules 2 and 3? Well, module 2 is looking at genomics and transcriptomics. So ChIP-seq is specifically a sequencing technique. You can map different segments across the genome, and using this you can see specific binding sites of a protein. This can be really useful information for looking at transcription factor binding, as I said earlier, nucleosome binding, all of which have impacts on gene expression. So that leads me to module 3, which was the quantitative analysis of gene expression. So as I said here, the transcription factors are obviously very important in gene expression, as well as histone modifications. So some of these modifications can impact chromatin accessibility and therefore impact gene expression. Some limitations of ChIP-seq. Number one, it requires a specific antibody for your protein of interest. So if you are studying a new protein that may not have a good antibody yet, this probably wouldn't be a good option for you. It needs to be a very specific antibody, so only binding to your protein of interest. That way you're not getting signals from other proteins that are binding to the DNA. So, as I mentioned earlier, the formaldehyde is very harsh, can lead to some artifacts such as preventing antibody recognition, preventing chromatin fragmentation, and even degradation of your sample. So this is something that is one minor characteristic of ChIP-seq. And next, you must have a reference genome to map your reads to. So if you're working with a new organism or a new sample that doesn't have a reference genome, then you may not be able to actually map these protein associations in your genome. Last, it is very expensive and time-consuming, so depending on your options there, this may not be such a good assay for you. So the advantages compared to other techniques, so as I said, ChIP-seq is a little bit of an older method. The only one that is older than ChIP-seq for this type of study is ChIP, which is the mapping of protein DNA interactions on a microarray, so a grid of DNA segments. ChIP-seq is preferred over this because it gives you single nucleotide resolution rather than just a 30 to 100 base pair range that is given in ChIP. Also in ChIP-seq, you need less DNA sample, which is nice. You can multiplex, get a lot more data at one time, and there's a better signal-to-noise ratio in ChIP-seq. So for your controls, you would want to have a sample with no antibody added, or a sample with an antibody that doesn't recognize your protein of interest, such as like a housekeeping gene or something like this. This can be used to assess your background signal levels. So this will tell you basically what amount of your signal is due to background levels or non-specific antibody binding. So to show you some ChIP-seq data in papers, this group is looking at nucleosome positioning and histone methylation across chromosome 21. So you can see here these peaks. This would be representative of where nucleosomes are and where histone methylation is across this chromosome. And they're also looking at CTCF, which is binding to the DNA and causing looping or topologically associated domains, which can impact gene expression. So here's just a nice picture of how ChIP-seq can help you to map different things on the genome. Next, this manuscript paper has looked at different protein binding sites to show associations between proteins. So here they're looking at the MIP oncoprotein transcription factor and WDR5, which is a cofactor that can recruit MIP. So here, very interesting, they see that at this point in the genome there's co-localization of MIP and WDR5, which showed that there is some protein association and functional relationship between these two proteins. This could be a potential target for therapeutic strategy and provides some information about how MIP is functioning as an oncogene. Last, this paper has looked at mapping histone modifications in relation to transcription start sites. So here they're looking at the acetylation of H4 histone and specifically looking at where it is localizing in reference to the transcription start site. So specifically here, it is important to know that acetylation opens chromatin, which allows more transcription activity. So here they show that most H4 acetylation actually occurs right near the transcription start site, meaning here that there's more accessibility of the chromatin, so transcription can actually start, transcription machinery can come in and start the process. They are looking here at the impact of BRCA2 loss, and they see here in red that without BRCA2 you actually have more acetylation, so this loss generates more acetylation of the histone, which in turn will result in more transcription of the genes in this region. So that is all for ChIP-seq. I hope you've learned something and have enjoyed this presentation. Thank you.